This article, Closure of the Neuro-central synchondrosis and Other Physes in Foal Cervical Spines, delves into the intricate growth patterns of foal cervical spines. The study centers around a crucial growth plate known as the neuro-central synchondrosis, NCS, responsible for the development of the upper third of the vertebral body and the protective neural arches surrounding the spinal cord. This research stems from observations in pigs, where hindering the NCS led to spinal stenosis, a condition characterized by a narrowing of the spinal canal that can potentially compress the spinal cord. Notably, horses also experience spinal stenosis, particularly young horses affected by wobbler syndrome. This study meticulously describes the NCS in foals, aiming to elucidate its contribution to spinal stenosis in horses. The research employed a post-mortem cohort study design, encompassing 35 foals of diverse breeds and sexes, with ages ranging from 153 gestational days to 438 days old. Computed tomography (CT) scans provided detailed images of the foal cervical spines. Each physis, the growth region between bones, was meticulously scored from 0 to 6 based on its closure status. A score of 6 signified a fully open physis, indicative of active growth, while a score of 0 indicated complete closure, marking the cessation of growth. To streamline the analysis, physes with similar anatomical orientations were grouped together. Figure 2 visually illustrates this grouping scheme using images from a newborn foal. A transverse slice of the first cervical vertebra, C1, in Figure 2a highlights the dorsal physis and the left and right neurocentral synchondroses. Shifting to the third cervical vertebra, C3, Figure 2b, depicting a dorsal slice, demonstrates the independent scoring of the middle portion of the NCS, while its cranial and caudal portions are grouped with their corresponding vertebral body physes. Figure 2c, showcasing a sagittal slice of C3, further emphasizes this grouping strategy and the three-dimensional orientation of the NCS. Figure 2d provides a transverse slice of C3, mirroring the orientation of Figure 2a, and reveals the oblique trajectory of the mid-portion of the NCS. Lastly, Figure 2e displays a dorsal slice of the second cervical vertebra, C2, where 6 distinct physes are scored. Table 1 meticulously details the scoring system employed in the study, linking the observed characteristics of the physes with their corresponding scores and interpretations. The study's findings unveiled the NCS as a thin growth plate that significantly contributes to the growth of the vertebral body and neural arches in 3 dimensions: height, width, and length. A pivotal discovery was the complete closure of the middle portion of the NCS in all cervical vertebrae by 115 days of age. This finding implies that the middle portion of the NCS ceases to contribute to growth beyond this age. Figure 3 graphically depicts the relationship between vertebral length and physial closure score in relation to age, showcasing the increase in vertebral length and the decrease in physial closure score as foals mature. The researchers also observed that foals exhibit distinct early and late phases of maturation, with some growth plates completely closing during the early phase, while others commence development during the later phase. The discussion section of the article explores the broader implications of these findings, particularly their relevance to spinal stenosis in horses. The researchers acknowledge that the hospital-based nature of their study population might limit the generalizability of the findings to all foals. Further research encompassing normal foals is recommended to confirm the wider applicability of the results. The authors underscore the importance of considering both gestation length and postnatal age when analyzing physial closure. Variability in gestation length can impact closure times, emphasizing the need for a comprehensive approach to age assessment. The study's key finding, that equine cervical vertebrae mature in distinct early and late phases, mirroring the growth patterns observed in children, is further emphasized in the discussion. The researchers also note the diverse developmental pathways of various vertebral processes, such as the articular and transverse processes. These variations in development could influence their susceptibility to disease, suggesting a link between developmental patterns and disease predisposition. The article concludes by reaffirming the crucial role of the NCS in cervical vertebrae growth and its potential involvement in the development of spinal stenosis. The authors recognize the need for continued research to fully unravel the complex relationship between the NCS and spinal stenosis in horses. They propose that disruptions in the blood supply to the NCS, particularly its cranial and caudal portions, might lead to delayed growth and stenosis in these specific areas of the spinal canal. Another hypothesis put forth suggests that osteochondrosis, a developmental disorder affecting bone and cartilage, occurring in the facet joints, could interact with NCS growth to further contribute to stenosis. This study serves as a foundation for future research on spinal stenosis in horses by providing a detailed understanding of cervical spine growth patterns, with a particular focus on the NCS. This knowledge will be instrumental in unraveling the complexities of the condition's development, paving the way for the development of effective preventive and treatment strategies. From Vet Neuro Jar, keep those minds inspired, hearts light, and tails wagging.